Hello, good day. Our lesson for today is Chromosome Variation. Imagine opening a book where every page holds the instructions for life, but suddenly, a few pages are missing, duplicated, or rearranged. That's what happens in chromosomal variation. Today, we'll explore how these tiny twists in our chromosomes can lead to fascinating diversity and sometimes serious consequences in living organisms. In this lesson, we'll define important terminologies in chromosomal mutations, differentiate among several types of ploidy, appreciate the importance of disjunction in the process of meiosis, distinguish between chromosomal deletion and duplication. The case of Down syndrome. Down syndrome occurs because there are three copies of certain genes located on chromosome 21. Researchers have identified a specific area, roughly 5 million base pairs long, that seems especially important. This region contains 33 genes. However, scientists are still working to understand exactly how these genes contribute to the condition. What do we know is that it likely involves complex interactions among multiple genes on chromosomes 21. On the topic of genes and chromosomes, we must first learn how chromosome is structured in each cell. So now that we've looked at chromosome 21 and its link to Down syndrome, let's take a step back and look at the structure of chromosomes in general. Understanding the basic parts, like the centromere and the chromosome arms, will help us see how genetic material is organized and why certain regions like Down syndrome critical region are so important. The smaller part of the chromosomes, the upper parts of chromosome, are called the P arms or short arm. Letter P stands for petite, petite meaning short. It is located above the centromere. On the lower part below the centromere, the arms are called Q arm or long arm. That is located below the centromere. Now that now that we know what the centromere is and where it's located, let's see how its position actually affects the shape of morphology of the chromosome. So when we speak of chromosome morphology, we are just simply talking about the position of the centromere on the chromosome. Chromosomes are classified based on the position of the centromere, like metacentric. When the centromere is right in the middle, we call it metacentric. When the centromere is in a bit of center, that's what we call as submetacentric. When the centromere is close to one end, it's acrocentric. And if it's at the very end, that's called telocentric. These differences in centromere position help scientists identify and categorize chromosomes more precisely. You are seeing right now is the karyotype of humans. The complete set of chromosomes is what we call as karyotype. Karyotype is arranged from the largest pair of chromosome up to the smallest pair of chromosome and the pair of sex chromosomes. For example, this karyotype belongs to a female since there are two X's for the sex chromosome. If you notice, the largest chromosome is chromosome pair number 1, and the smallest chromosome is the chromosome pair number 22. Now that we understand how chromosomes differ in shape and structure, let's move on to what happens when the structure changes. These changes are known as chromosome mutations. There are several types of chromosome mutations. The main categories include number 1, rearrangements, which involve changes in the structure of a chromosome. Next is aneuploidy, where the number of chromosomes changes, either missing or extra copies, and polyploidy, where there are whole extra sets of chromosomes. Each of these mutations can have major effects on how genes are expressed and how an organism develops. So this is just a diagram showing you the differences of the chromosomal mutations that we just mentioned. In duplication, there is a doubling of a part of a chromosome. It could be tandem duplication, where the two consecutive genes are being repeated in one arm, or displaced duplication, where the two consecutive genes are placed within the same arm, but there are other genes in between them or two other chromosomes, and the reverse duplication, where the two consecutive genes are flipped in order to produce a reverse sequence, but they are near to the original or close, right exactly beside the original sequence of genes. What do you think are the consequences of duplication in an organism? To better understand how chromosomes mutation affect physical traits, let's look at the classic example, the bar mutation in fruit flies. The bar mutation reduces the number of facets in the fly's compound eyes. As a result, heterozygous females and hemizygous males that carry this mutation have noticeably smaller eyes. This example, as you can see here, shows how even a small change in chromosome or chromosome structure can produce visible, measurable differences in an organism. The doubling of a segment of a chromosome will produce drastic changes in the facets of the eyes of the Drosophila. So how does the duplication affect phenotype? Well, there are many effects. First one, there would be unbalanced gene dosage. There would be interactions of many genes or gene products that can influence development. It could even produce novel functions. This is one of the ingredients in evolution. Example, the human globin genes, the segmental duplications on one chromosome, we call that intrachromosomal, or in different chromosomes, we call that interchromosomal. This picture shows you how the doubling of genes produce new phenotype in an organism. So we can see here, the doubling of B genes produce abnormal development in this species of fish. We've seen how structural changes like the bar mutation can affect an organism's traits. Now let's explore one specific type of structural change. We call this gene duplication and its broader consequences. In individuals heterozygous for chromosome duplication, the duplicated region of the chromosome loops out when homologous chromosomes pair in prophase 1 of meiosis. Duplications often have major effects on the phenotype, possibly by altering gene dosage. And segmental duplications are common within the human genome and have played an important role in the evolution of many eukaryotes. After looking at duplications, let's now examine another major type of chromosome rearrangement, deletions. While duplications add genetic material, deletions, on the other hand, remove it, often with more serious consequences as compared to duplications. The effects depend on whether the individual is homozygous or heterozygous for the deletion. If homozygous or in homozygous condition, deletions are usually lethal because there are two pairs of exact chromosome or genes. In heterozygous conditions or individuals, deletions can cause genetic imbalance, pseudodominance, or reveal haploinsufficient genes or genes that require both copies to function properly. This is a picture this is a picture showing you the formation of deletion loop during pairing of homologous or homologs in prophase 1. The appearance would be just like a kind of a loop. You may pause this video to read the different types or the different effects of human chromosome rearrangement, from duplication to deletion. After deletions, another important structural change we can observe in chromosomes is called an inversion. Unlike deletions, no genetic material is lost, but the order of genes is rearranged. Inversions is when a chromosome segment is inverted or turned 180 degrees. Depending on whether the centromere is included, there are two main types. One type is paracentric inversion, where the centromere is not involved, as in the case of AB. If you see the symbol, a little circle there in after the B or before the C, that is the symbol for centromere. AB centromere, CDEFG. So these are the sequences. This is the sequence of the genes, and there is an inversion happen in the sequence of DEF genes, so it became FAD without the involvement of the centromere. In a pericentric inversion, the centromere is included this time, as in the case of AB centromere CDEFG. There was inversion, and this became ADC centromere BEFG. Even though no DNA is gained or lost, inversions can still cause significant effects. They may result in a gene break, splitting one gene into two parts, or a position effect, where the expression of a gene changes because it's moved into a new locations. This is another example of the two types of inversion, paracentric and pericentric. Para means next to the centromere. Peri means around or around the centromere. Inversions can cause breaks in some genes and may relocate others to entirely new positions in the chromosomes. In individual heterozygous for an inversion, the homologous chromosomes must form an inversion loop during prophase 1 of meiosis to align properly. However, when crossing over happens within this inverted region, it usually produces non-viable gametes. As a result, we often see a decrease in recombination frequency. This is a phenomenon known as inversion suppression. The other type of chromosomal mutation is translocation. Translocation is the movement of genetic material between non-homologous chromosomes. There are many examples of this, but mainly we will be discussing two main examples of translocation. One is non-reciprocal translocation. Non-reciprocal translocation is when one chromosome transfers a segment to another chromosome without receiving anything back, and that's why it is called non-reciprocal. As for the example, AB centromere CDEFG, and the other chromosome, a non-homologous chromosome, MN centromere OPQRS, leads to AB centromere CDG, MN centromere OPEFQRS. You've seen that the genes EF are now transferred to the MN centromere OPQRS chromosome. There's also reciprocal translocation. This is when both chromosomes exchange segments with each other, as reflected in this example. These translocations can have several consequences. They may create fused genes that produce fusion proteins. They can cause position effect by altering gene expression, or they might break within a gene, disrupting its function. A special form called a Robertsonian translocation occurs when two acrocentric chromosomes fuse at their centromeres, forming one large chromosome, and often leading to conditions like certain forms of Down syndrome. This is how a reciprocal translocation arises. Two normal pairs of chromosomes in the left-hand side of the picture. In the middle, parts of two chromosomes break off, and then they attach to different chromosomes. Remember, translocations happen between two non-homologous chromosomes. In non-reciprocal translocation, genetic material moves from one chromosome only to another without any reciprocal exchange. In Robertsonian translocation, one short arm of one acrocentric chromosome, it is exchanged with the long arm of another, creating a large metacentric chromosomes. And some of the cases of Down syndromes are formed through Robertsonian translocation. After discussing major chromosomal rearrangements, like translocations in the previous slide, let's look at a more subtle form of chromosome instability, something known as fragile sites. Fragile sites are specific areas in chromosomes that appear as constrictions or gaps, and are prone to breakage under certain conditions. A well-known example is Fragile X syndrome, a genetic disorder associated with intellectual disability. It occurs in about 1 in every 5,000 male births and is caused by an increase in the number of CGG trinucleotide repeats within a particular gene on the X chromosome. In this picture, shown here is a fragile site on human chromosome X. Finally, let's look at a broader category of genetic variation that occurs naturally in many people. These are called copy number variations, or CNVs. Copy number variations refer to differences in the number of copies of particular DNA sequences among individuals. They often involve duplications or deletions that can range from a few thousand to several million base pairs in length. Interestingly, CNVs are quite common in the human genome and have been linked to a variety of traits and conditions, including unusual intelligence, osteoporosis, autism, schizophrenia, and several other diseases and disorders. Now let's take a look at a different kind of mutation that involves the whole chromosome itself, or the number chromosome. A good example would be aneuploidy. Aneuploidy is the loss or gain of one or more individual chromosomes. It disrupts gene dosage and often has severe phenotypic effects. There are many causes of aneuploidy. It could be from the deletion of centromere during cell division, Robertsonian translocation, or non-disjunction during meiosis and mitosis. Especially non-disjunction, where two homologous chromosomes did not separate, and as a consequence, they are brought into the new daughter cells, forming an imbalanced number of chromosomes in the daughter cells. There are many types of aneuploidy. One is nullisomy, when there is a loss of both members of a homologous pair of chromosomes. So that would mean a diploid number minus 2 chromosomes. Monosomy involves loss of a single chromosome. So that would mean diploid number minus 1. Trisomy gain, it is a gain of single chromosome. That is diploid plus 1 extra chromosome. Tetrasomy gains 2 homologous chromosomes, diploid plus 2. And there are more examples. Examples: the 2n plus 1 plus 1, or the double trisomy, 2n minus 1 minus 1, or double monosomy, 2n plus 2 plus 2, or the double tetrasomy. There are many effects of aneuploidy. There are mostly drastic effects. There could be gene dosage imbalance. And if it happens in humans, it can involve sex chromosome. For example, as in the case of Turner syndrome, where there is only one X chromosome in females, and Klinefelter syndrome when there is an extra X chromosome in males. If it happens in autosomes, it could include disorders like trisomy 21 or the Down syndrome, and we've mentioned it before that there are many types of Down syndrome. The first one is primary Down syndrome, where it results from 75% random non-disjunction in egg formation, or the familial Down syndrome. This is a kind of Robertsonian translocation between chromosome 14 and 21. In short, there is an exchange of segments of chromosome 14 and 21. This figure shows translocation carriers are at increased risk for producing children with Down syndrome. Now that we've explored structural changes in chromosomes, let's shift to numerical changes, specifically in humans, aneuploid in humans. In humans, autosomal aneuploidy occurs when there is an extra missing copy of one of the autosomes. Example, trisomy 18 is known as Edwards syndrome, which occurs in about 1 in 8,000 live births. Trisomy 13, or the Patau syndrome, it happens in 1 in 15,000 live births. And trisomy 8, often seen as mosaicism syndrome, appearing roughly 1 in 25,000 or 1 in 50,000 live births. Why is there a drastic decrease in frequency of these trisomic syndromes from chromosome 18 to chromosome 8? You might notice that there is a drastic decrease because this happens because larger or gene-rich chromosomes can't tolerate extra genetic material as easily. So in most cases, these imbalances are lethal during development. So only a few specific trisomies are seen in live births. This is an example of a baby with trisomy 18. This is trisomy 13 or Patau syndrome. The incidence of primary Down syndrome and other aneuploidies increases with maternal age. In other words, older mothers are more likely to give birth to a child with Down syndrome compared to younger mothers, as in the case in this graph. So the older the mother, the more likely that she will give birth to a child with Down syndrome. There is also what we call as polyploidy, and this is the presence of extra chromosomes or chromosomal sets. This is common in plants and less so in animals. The cause is a failure of whole sets of chromosomes to separate. There are types according to the number of sets of chromosomes. For 3 sets, we call it triploidy. For 4 sets, tetraploidy. For 5 sets, that's pentaploidy. While aneuploidy involves changes in the number of individual chromosomes, sometimes entire sets of chromosomes are duplicated, and this is polyploidy. This is autopolyploidy, a special case of polyploidy. It occurs when accidents during mitosis or meiosis produce extra sets of chromosomes, all of which come from the same species. This type of chromosome duplication is fairly common in plants and can result in larger cell size, bigger fruits or flowers, and sometimes reproductive isolation, contributing to the formation of new species. As you can see, there could be unbalanced chromosomes after meiosis of a triploid cell. Now that we've seen how extra chromosome sets can arise with a single species, let's look at what happens when 2 different species are involved. When 2 different species are involved, we call this allopolyploidy. Allopolyploidy results from hybridization between 2 species. The hybrid ends up carrying extra sets of chromosomes, but this time, those sets come from two or more different species. Interestingly, many allopolyploids are fertile. In some cases, hybrids chromosomes double during mitosis, producing what's known as amphidiploid. This is just a tetraploid organism that restores proper pairing during meiosis. In short, allopolyploidy is an important mechanism for speciation evolution, especially in plants. So, the significance of polyploidy. Polyploidy is a major contributor to evolutionary success in several groups. Vertebrate genome has duplicated twice, once in the common ancestor of jawed vertebrates and again in ancestor fish. Nuclear volume increases, thus cell volume increases. A selection of plants with bigger leaves, roots, flowers, and seeds, and new and extra copies of genes give rise to new functions. New and extra sets of genes may give rise to new species. Now, because of polyploidy, many of the food that we're eating, plant material, are actually polyploid. Potato is a polyploid, banana, peanut, sweet potato, tobacco, cotton, wheat, oats, sugarcane, and strawberry are all polyploid organisms. They may be resulting organisms from autopolyploidy or allopolyploidy. You may pause this video in order to study this table showing different types of chromosome mutations.